Hello, I hope you're doing well. This is Arshak Rezvani, and today I will be presenting our paper accepted to Actionable Interpretability Workshop at the International Conference on Machine Learning 2025, which is titled Interpretable EEG to Image Generation with Semantic Prompts. The contents of this representation will be some introduction and the definition of the core problem. Then I'll go into our methods. Then I'll introduce you to our results. And lastly, I'll go over some interpretability takes on our model. And lastly, I'll conclude. So the goal of this paper is to reconstruct a visual perception from EEG recordings, which has a high value for neuroscientists and brain-computer interfaces and the ML literature itself. EEG is low-cost, non-invasive, and has high temporal resolution, which makes it suitable for many clinical settings. And that's why we chose this modality. And the challenge of this modality is that it suffers from low SNR and it has limited spatial resolution, which affects the coherency and the quality of the generated images that we try to overcome in this paper. So in our methods, instead of directly reconstructing the images from EEG, we use text as a mediating medium between the two modalities being image and the EEG recordings. So we have an image data set during train time that we will ask an LLM to semantically describe these images using 10 prompts that vary from low level, mid level, and high level. And after getting the answer to this question, which are the semantic captions, we have a text encoder, which is pre-trained, that will give us a EOS token, end of sentence token, that describes the caption per semantic level. And then we have a transformer-based neural network that we call EEG encoder that has 10 heads, that each head will give us a vector which represents the 10 semantic levels, and we have 10 semantic captions embedding as well. So we will align these 10 captions with these 10 EEG embeddings using some contrastive learning loss here. And we'll optimize our model using typical optimization tools to train the EEG encoder. And after training that, during the inference, we take a new EEG and we feed it into our EEG encoder, and we will get 10 representations. And per head representation, we'll do a search in that semantic level data set through all the captions, and we find the top pair per semantic level. So we will get 10 captions that will semantically describe the image, that hopefully will semantically describe the image, and we will select the top ones that had the high similarity score, and then we will concat them and give them to a stable diffusion, which is a text to image pipeline, and we will get a plausible image of the EEG activity. This way of training the model will enable us to do some interpretability analysis on our model, which I'll go into details in the next slides. So during the train time, we have some prompts that vary from low level, mid level to high level. One example of the low level prompt would be what objects are present in the image. One example of the mid level prompt would be what is the spatial relationship between the main objects. For example, is something next to, below, or above something. And lastly are the high level prompts, which say what emotions or moods does image convey, or what is the theme of the image. And we will ask these questions paired with the image from an LLM. In our case, it would be GT 4.1 mini. So we will get a semantic data set, which we will use to train our model and reconstruct the images from the EEG. During the inference, we try to find the captions using this. So each input is mapped to the most relevant caption via the trained encoder. Per head, we try to do this, solve this equation here. So per head, we have a caption data set. Per semantic level, we have a caption data set. And if we denote our EEG a test time EEG as X test, and the encoder of text and EEG as F text and FEEG, we can, and sim would be the similarity score, which is the cosine similarity in this case, in our case, and we have a superscript index called k, which refers to the semantic level. For example, object details, spatial layout, or mood. So per semantic level, we will find the caption which is closest to our EEG, and we will call that C hat, and we will choose the ones that get the highest score, and then we will concatenate them and give them to a text to image pipeline. So here are some results. We worked on EEG CVPR dataset, which is an EEG dataset paired with some images, which is a subset of the ImageNet dataset. And we computed some metrics, and we get the highest score on all metrics compared to other works. And we also computed some other metrics that weren't relevant in this type of literature, but were similar, metrics that were similarity between the distribution of the reconstructed image and the original stimuli. And here are some results which are visual. At the top, we can see the original image. And in the bottom, we can see the reconstructed image from the EEG. For example, here the image that the person saw was a pair of shoes, a pair of Nike shoes, which we could reconstruct the Nike logo as well. And as you can see, the results are robust, and we were able to reconstruct the visual stimuli pretty well. So for the interpretability analysis, we had for each head, we had a neural network that we tried to compute the input gradient of the input with respect to output loss, which will give us a matrix which is the same shape as the EEG. So we averaged this across all the data set, and this gave us one singular EEG activity across time and electrodes. And then we computed the average across the time dimension. So we get the activity across the electrodes, and we plotted them using the topomap plotting tools, and we got these images per head. So as you can see, most of the activity is in occipital regions because the stimuli is aligned with the typical knowledge in the neuroscience field. And for some high-level features, we can see some activity in frontal regions, which also agrees with the literature in the neuroscience. Secondly, we computed the TSNE of the EEG encoder outputs, and we saw that each head is specialized in some part of the latent space. So we have per head, we have a vector, and then we reduced the dimension of this vector to 2 using TSNE method, and then we colored based on what head that embedding was. As you can see, the heads are distinct, and they're describing different parts of the latent space, and that's why they enable us to reconstruct the image very well. Lastly, we try to find which heads are most important by computing the highest similarity score and counting which head had the high similarity score and plotting the histogram of it. And as you can see, the importance of the object snap prompt, which asks what objects are present in the image, is the highest, and so on. And for conclusion, we introduced the novel EEG framework, EEG to image framework, that uses multi-level semantic prompts. And we also achieved state-of-the-art results on the EEG CVPR dataset across multiple metrics, and we demonstrated that each EEG head is specialized in a distinct semantic level. And we also did some interpretability analysis using gradient-based saliency maps. This work could be applied to clinical settings because it's pretty lightweighted and requires limited training. And here are some other images which are reconstructed. Here is the real images, and these are the reconstructed images, as you can see. And lastly, this is our team. I'm Arshak Rezvani, and thank you for your attention.